I'm Tim Ventura, and we're joined today by Dr. Stephen Benner, known for his work in synthetic biology, including creating in the lab DNA that might be found in alien life forms, Jurassic Park experiments that resurrect proteins from ancient Earth life, and generating COVID test kits that were used in India, Europe, and the US, among many other contributions to science. Dr. Benner has been a professor at Harvard University, ETH Zurich, and the University of Florida, where he was a VT and Lewis Jackson Distinguished Professor of Chemistry. In 2005, he founded the Westheimer Institute of Science and Technology and the Foundation for Applied Molecular Evolution. Dr. Benner has also founded the companies EraGen Biosciences and Firebird BioMolecular Sciences. Dr. Benner and his colleagues were the first to synthesize a gene, beginning the field of synthetic biology. He was instrumental in establishing the field of paleogenetics. He's interested in the origin of life and the chemical conditions and processes needed to produce RNA. He's worked with NASA to develop detectors for alien genetic materials using the definition of life developed by the NASA Exobiology Discipline Working Group in 1992, a self-sustaining chemical system capable of Darwinian evolution. Dr. Benner has a BA and MS in molecular biophysics and biochemistry from Yale and a PhD in chemistry from Harvard, and has been awarded over a dozen awards and honors related to his research. So, Stephen, welcome, sir. It is truly an honor to have you with me here today.
Well, the honor is all mine. It's good to be here.
Well, so today we are talking about, I mean, you have done remarkable work across a number of disciplines, but I wanted to try and focus on the field of synthetic biology. Really, that, for me, when I read about that, I was just blown away. I had no idea that this was being done and it had come so far. So you are basically described as the founder of this field, right?
Yeah, I mean, there's a number of disciplines of synthetic biology that go in different directions. The one part of it that we are associated with the origination of is the ability to rebuild living systems from the bottom up. How do you create artificial cells? How would you do genetic molecules differently than DNA? Keep in mind that on Earth right now, you are looking, even though you have plants and animals and bacteria and so on, you're really looking at only one example of life. It's all descended from a common ancestor, at least the life that we know on Earth. And therefore, it all has very much the same biochemistry. And so one of the questions of, it's a challenge of synthesis is that you don't understand something unless you can make it, and especially if you can make it in a different form. So what we do is try to create different kinds of genetic molecules so that we can understand the molecule, genetic molecules that we have, DNA and RNA in particular. And by doing so, you can create molecules that may very well have emerged naturally in other planets with other natural histories. So that's the part of synthetic biology that we're associated with.
Again, these are tremendous ideas. I mean, this research just blows me away. So I want to get into Hachimoji DNA. I hope I'm pronouncing that right. And that's a synthetic nucleic acid analog that uses 4 synthetic nucleotides in addition to the 4 natural ones, leading to 4 allowed base pairs. So it has 2 normal base pairs, right, that normal DNA does, and then it has 2 unnatural base pairs, which are synthetic. And so, again, the thing that's exciting about this, and I'm worried that I may lose the audience a little bit, right, because we're digging in here. But so with DNA, you normally have 4 base pairs. And in this case, you're creating 2 new ones that have never existed in nature, right?
Right. Well, let's try to not lose the audience. So the lead scientist on this paper was a fellow named Shuichi Hoshika, who is actually Japanese, and then Nicole Leal, who is American. But the Japanese word for 8 is hachi, and the Japanese word for letter is moji, which you may know as emoji, which is an emotional letter. So Hachimoji is 8-letter DNA. And as you mentioned, right, the DNA that you and I share, and as far as we know, all life on Earth shares, has just 4 letters in the genetic alphabet. And these are G, A, C, and T. And so all of your genetic information is written with just those 4 letters. But when you ask the question, what if, or why not, right, you can actually draw out, as a chemist does, on paper, other ways of having two Lego blocks fit together with prongs and holes. And it turns out that with A, T, and GC, which are the base pairs that you have in your genome, the prongs and holes are not completely used up. We can have other combinations of Lego bricks that fit together. And of course, as a chemist, you can actually synthesize these in the laboratory. So we can have actually as many as 12. The paper that you're referring to is, I guess, from 4 years ago, which just looked at 8 of them working together. So you have 8 different letters, which in fact, for those of you who are into computer science, right, if I just 2 letters, I have 1 bit of information every site. 4 letters, I have 2 bits of information at every site. So 8 letters, I have 3 bits of information at every site. 16, I would have 4 bits of letter information at every site. So it goes with a power. But yeah, so this is DNA that has 8 different letters. It can encode more amino acids than the 20 amino acids that you and I have in our proteins, as well as all other life on Earth. It can form stable structures. In fact, one of our big diagnostics kits uses these to measure viral loads in patients infected with HIV and hepatitis C, for example. It's also been used various other diagnostics tests. But yeah, so you got your own DNA, your own molecular recognition system, and you can play around in the lab, and it does all sorts of useful things.
Well, so let me get into some of the applications for this, which were listed as developing clean diagnostics for human diseases, which you just mentioned, DNA digital data storage, which you touched on, DNA barcoding, self-assembling nanostructures, and making proteins with unusual amino acids, which again, you mentioned there were far more than the 20 that we see in nature. So in a simplistic sense, one of the key applications for this technology seems to be encoding digital information into the DNA of a living system, right?
Exactly. Well, that's right. So keep in mind that this is sometimes we call these semi-synthetic organisms. There's a gentleman named Floyd Romersberg who's at a company called Synthorx, who actually has done this as well. But the idea, of course, is to have additional information density within the system. It's also what we like to call orthogonal density information, which is hard to explain to the layperson without a lot of pictures. But the idea is pretty simple. If I have AT and GC as my 4 letters in my DNA that I'm using, and I want to go into a living system, and I try to use my own DNA with AT, GC, and add synthetic DNA with AT, G, and C, the DNA that's in my body with AT, G, and C can interfere with what probes I might add or what reagents I might put into my body to detect a virus of some kind. And so what we are doing is using additional letters, ZP, KX, SB as pairs to let us do things inside of a living system without being interfered with and without interfering with what's going on in that living system with AT, G, and C. So this idea of having a separate set of information molecules that you can associate with a living system, which is using a different set of information molecules. I can have information here that does not interfere with the living system's information. And of course, the information that which we're adding is not being interfered with by the information from the living system. So these are tools that you can do to go in and now do clean diagnostics, for example, where you're using the information that we are adding by synthesis to signal the presence or absence of a natural DNA like HIV or hepatitis C, and able to do so without the two systems interfering with each other.
So it sounds like with these additional letters, they can be used to synthesize proteins, right? So if you encode this into the cell, even though it's primarily used for data storage, you could also use this to program, again, you'd mentioned creating new proteins. But one thing that I wanted to mention was currently this system is not self-sustaining because it requires unique building blocks that aren't produced in nature, right? That's going to keep that's our sanity check for the audience. So there's no Jurassic Park here because they require proteins that aren't found in nature. So there's no way around that.
Well, that's right. So we actually encoded extra amino acids in the protein lexicon, shall we be linguistic in our analogy here, using extra letters in the DNA alphabet all the way back in 1991. Floyd Romersberg, actually, who I've already mentioned, has actually done the same thing with extra letters in living bacterial cells. But you're right; these are systems that are not self-sustaining. In fact, we don't try to make them self-sustaining, because there's a biohazard question here, which we
Yeah.
want to steer very much clear of. But these are systems, in fact, that are so orthogonal, again, there's the word, not intervening with each other, that not only do they not have the components that they need to survive unless we give it to them, but also they don't eat you. They don't want to eat you because you don't have the components that are necessary for them to survive. So this is the general argument is that I'm not encouraging cannibalism here, but the most nutritious things that give you the building blocks that you most need are your nearest animal relatives, like steak and chicken. But the more distant you get, the less likely you are to find it tasty. So, for example, if you eat Martians, that's not likely to be very nutritive, because the Martians are not very much like you, assuming that they have not shared a natural history with you.
Okay. Well, so I want to touch on, I guess, a more expansive scope here, because Hachimoji DNA is actually related, I believe, in some ways, to something called AEGIS, the Artificially Expanded Genetic Information System. This was a precursor, and it used 12 different nucleotides, including the 4 found in DNA. It sounds like that was larger and more expansive, and yet you had developed that before Hachimoji.
Yes.
Was there a reason why you guys trimmed that down?
Yeah, absolutely. So the 12 is actually, given general rules of chemistry, the most that you can have. There's an argument for why that's the case, which I will not go into. But now, of course, you've got to get them working together. You've got to get enzymes that will copy the DNA that you've added letters to. You have to get, of course, we have a local manufacturing operation here at Firebird BioMolecular Sciences that is making these things in large amounts so that we can make them. And so at some level, you have to decide how many more do you need to have in order to do things that you want to do with them. So, of course, the initial applications were with 6. We picked 2 out of the extra 8 to add to the 4 that are natural. So 2 plus 4 makes 6. And so that's what's going into diagnostics kits and all the rest of it. The 8 is what we really want to do if we want to have a more expansive evolution system, so that we can create drugs that bind to cancer cells and kill them and this type of thing. But of course, as you perhaps have guessed, I have many ways of getting 8, right? I got 1, 2, 3, 4 from nature, and then I got 5, 6, 7, 8. But of course, I got a 1, 2, 3, 4 plus 9, 10, 11, 12. And so we have to come up with a different 8. So I've got a nice Korean language speaker who will do the same thing. Instead of in Japanese, Hachimoji, we'll do it in Korean, and then we'll do Spanish at some point. So there are many mixes and matches. But if you can get away with doing 8, it saves you all the work of making the 9th and the 10th in the laboratory, or number 11 and number 12, and doing all the analytical chemistry and all the synthetic chemistry. Remember, each one of these things, not only do you have to make them, but you have to know that you've made them. And that's what we call analytical chemistry. That means you have to take what you have made and then figure out what the atoms are in it and how it's bound and put together. And that's, of course, itself a project. So 8 is good enough for us to do the next level, although we have done 10. Okay, so you have to pick your language, maybe Romanian, and then we can go and give the name to that alphabet. But that's why we do 8. It's just as a matter of practical use. It just,
Okay.
if you don't need more than 8, we don't go to the effort of using more than 8.
That makes sense. It sounds like complexity is a major factor for that. Well, so speaking of complexity, synthetic biology in general strikes me as being highly intersectional. I mean, you're not only working with genetics, biochemistry, and elements of traditional medicine, but also bioinformatics and information theory. And conceptually, this seems aligned with nanotechnology as well. So
Yes, absolutely.
does this interdisciplinary nature present challenges to doing the work, finding different resources, as well as finding team members who are able to just work across the expanse?
Well, yeah, it makes it fun. I mean, of course, people come here to learn interdisciplinarity, But you got to keep in mind, if you want to if you real interdisciplinary, we can talk about how these systems may have originated on Earth, because then you have to include planetary science and geology. So the synthetic biology, in some senses, rather small interdisciplinarity compared to what you would think of having to do if you wanted to talk about origins on a planet that is being bombarded by meteorites and having rocks bubble up from volcanoes and all the rest of it. So yeah, it's a challenge. It is, of course, a training opportunity for people in interdisciplinarity. But you got to remember, I mean, we try to train people to move smoothly from bioinformatics tools to synthetic chemistry, to biochemistry, enzymology, some physics, some biophysics, all the way over to living cells. And, 50 years ago, that would have been considered impossible, but not today. And part of the reason it's better today is because you can Google things. And, you're not perhaps as old as I am, but maybe you do remember going to a library. No, today's students don't go to libraries. They don't need to. And so it's become much, much easier because of technology, especially information technology, to be multidisciplinary.
Yeah, yeah. Actually, yeah, in my case, our university library, we had a card catalog on the bottom floor, and the physics and engineering texts were on the top floor. And so it used to be running up and down stairs, and then, I had like a change in my pocket for the photocopier, right?
Yes,
And so I would
exactly.
go home at the end of the day with, a stack of photocopies, and yeah.
Exactly.
So,
It was very hard to look things up.
oh yeah, it was. And it was hunting for the book, and was it misplaced or misshelved? And then you find the book and it doesn't have the information you need. So yeah.
Well, Robert Heinlein, the science fiction writer from the 1950s, said that all science begins with library science. It's just trying to understand, without a great deal of effort, what generations of people before you have learned, so you do not need to learn it all over again. But most of my son does not have a clue as to what a library is. He just never been through one of these card catalogs, which had filed 8.5 by 5-inch file cards, but that's one of the reasons why this multidisciplinarity is so easy. But it's also true for analysis, right? I mean, when I was a student, if you have a bottle of a chemical in it, you would spend a month or longer to figure out what the arrangements of carbon atoms and hydrogen atoms and oxygen atoms and nitrogen atoms were in that compound. Today, we can have the structure of the molecules in a bottle known by the afternoon.
Well, I wanted to jump into a couple of more speculative questions, I guess. And so one thing that I was just intrigued about was, has this work in synthetic biology revealed any particular advantages of natural DNA base pairs? I was wondering, are there any advantages? Again, there's A, C, G, and T. And I'm wondering, are these just the random product of evolution that's stuck because they locked in early, or are there actual advantages to those particular chemicals and the way that they're arranged?
That's the question, what if and why not? And we love these questions. So there are certain features of you as a living organism that are reflected adaptation for your current environment. There are certain features that are vestigial from times long ago, which you haven't bothered to change. There are certain features that are arising by random chance. So you have 5 fingers, right? Well, if you're a Simpsons, you have 4 fingers, but never mind. 5 fingers, why? Well, that's a decision that was made roughly at the boundary between the Triassic and the Permian, somewhere 250 million years ago, or a little bit longer. And this is fixed, right? So there's an interesting problem with information storage, right? Is that if I have just ones and zeros, that's a binary code like AT, right? What I would do is, of course, write information without a great deal of density. So the strings of ones and zeros is very, very long if you want to write a Shakespeare play. 26 letters is actually a very much better way of making compact information. But with 26 letters, you have to avoid misreading, right? So you see a 0 and an O, you got a problem, okay? That's, of course, the numbers and the letters are easily confused. And this is one of your major problems, is that if I want to copy things where I have to match an A with a T or a G with a C, There's the possibility of a mismatch between a G and a T, let's just say. And the more letters you have, the more mismatches you have. And the number of mismatches that you have scales roughly with a square of the number of letters. So there is likely an optimum. And that optimum is probably somewhere around 8. I have some colleagues, Westheimer, who is in Hungary, is a friend of mine. He says 4 is optimum. Of course, he knows that you and I have 4. So as we like to say, modeling is doomed to succeed. That is, if you do a theoretical study, you always get the answer that you know is in advance. And if you don't, you have to go back and do the theoretical study again. But the bottom line is that somewhere more and more letters leads to more and more mismatch potential, which then means to lower and lower fidelity, less faithful replication of the information. So there are arguments for this. And then there's the other possibility, you know, could these letters have emerged from pre-biological rock, earth, air, fire, and water in some scenario before life emerged? Now, of course, after life emerges, all sorts of complex molecules can evolve. Gramicidin, you know, cyclosporin for organ transplants, penicillin for disease infections. These are all products of Darwinian evolution, which is, of course, able to do things rationally. But before Darwinian evolution, you get A, you get G, you get C, you get U, that is uracil. You don't even get T, a different version. Now U pairs with A, so you get away with it. But the bottom line is that almost all of the molecules that we are using in the expanded DNA alphabets are made by processes that are not likely to have occurred on early Earth without life.
Ah, okay.
So A, T, G, and C looks like they're the vestiges of a time long past, the vestiges of pre-biological synthesis on Earth that was got rocks and all the rest of it, but also has had impacts from meteors that are reducing the atmosphere to allow hydrogen cyanide and various other compounds to be made that lead to these specific things, not Z and not P and not S and not B and all the other molecules that we have.
Well, it's remarkable to see, you know, to understand better where this all came from. And again, for me, one of the more remarkable things also is where it could go. I had read, I didn't put this into my questions list, but I'd read there were a couple of research scientists who did zip file encoding on DNA. And they apparently they had said something, and again, I don't have notes on this, but they'd said that traditional zip file encoding didn't work well, but if they translated the way that they did that, they were able to make that work effectively. Then I mentioned that to a friend the other day, and he said instead of zip file encoding, they should look at error correction, right, for space travel. And he was like, you could put error correction algorithms in there. And as I understand, DNA already has some of that in there, but.
Well, yeah, I mean, keep in mind that your flash drive in itself is a work of engineering marvels, right? You're a futurist, so you understand that. Well, you've lived in the past, right? So you know that that kind of technology where I take a little stick that roughly the size of my thumb, it's a thumb drive, and put it in and have, you know, 14, 16 gigabytes of memory and instantly available to me. If I had mentioned this to you in 1970, you would say, it can't be done, right? This is, I mean,
Oh,
would,
yeah.
then your physics brain would have turned on. You would have started to think about what had to be true, or what has to happen for it to be done. But that's right. So the IBM is a team in Seattle. IBM has been working on trying to write DNA information in DNA. You do it. I mean, you've got 3 billion base pairs in your body right now. The reading and writing of it is a little slower. I mean, you got to keep in mind that if you watch the developments of chips and thumb drives and all the rest of it, the reading and writing has gotten extremely fast. And so the problem with DNA is that you are reading it and writing it slowly. Now, one of your problems is if I have a pot, I can put your DNA into a pot this big and have all the information in your body. Now, to retrieve it I had to put a probe in. So if I want to retrieve A, I got to put in a probe which has T. Well, that probe takes a lot of time to find the A, because sometimes it'll find AAGAA, and they'll say, oh, this sounds pretty good. So it'll stick to it even though there's a mismatch, but it will spend time sticking to it. So the rate at which you can find information out of a pot of 4-letter DNA can be quite slow. But make it 8-letter DNA, and now there's a lot faster searching for that information because there's a lot more information density, which means there are fewer places that A can get distracted, because there's not likely, we won't put into there AATAA, we will make it AAPZS, right? Got extra letters, we will use them to increase the information density. But yeah, so your problem right now is that your competing technology, which is what we're operating on right now, right, is not, it's just so good. Now, keep in mind, there are cases where you want to store DNA for a long time, where you're not going to look at it very frequently. So, for example, say you're looking with the space telescope and you're looking at a quadrant of the sky, you're collecting an enormous amount of information in an evening. Now, you might look at it as it's coming in, but then say 10 years, but then you'll store it in DNA, and then say 10 years later, there'll be a supernova in that region of space. So you will want to go back to what you stored in DNA 10 years earlier and look at that specific region. Another thing is, if you go to a medical doctor, you go in, they'll take x-rays, body scans, an enormous amount of information. They'll look at it in real time, but then you will want to store it for a longer period of time. And then 10 years from now, if you have pancreatic cancer, they might want to go back and see what your body was doing in that part 10 years ago, when it was not, when the data were first collected. So there are applications for slow read, contemporary write applications, and those are the kind of things that DNA, especially when you have extra letters in the genetic alphabet, can help you solve.
Yeah. Well, and again, this is absolutely brilliant. I mean, just being able to look at DNA and biological processes, again, from an information theory perspective, right? And being able to put that into kind of that IT context that we're all familiar with now through computers. It's just breathtaking. So NASA funded this research as part of an effort to, quote unquote, expand the scope of structures that we might encounter as we search for life in the cosmos. And from what you've already described, that makes absolute sense. It sounds like NASA was basically wondering how DNA or reproductive information systems on other worlds might be different than ours. Would that be kind of where they're coming from?
You got to keep in mind that there's a history here, right? And as you know, the most important thing in science is money, and no buck Rogers is, I think, the line from Tom Wolfe. And so, yeah, so when I was in 1986, the reason why I moved to Switzerland was because this whole thing was a crackpot idea. No one would fund it. We moved to Switzerland because when you're a professor at the ETH, as you mentioned, the Eidgenössische Technische Hochschule, you are given a lot more flexibility and funding, and a lot less peer review. So, I mean, peers can kill anything that is past the pale, beyond the pale, into the area of innovation. I mean, forget futurism here. We're talking about next dayism will kill a grant application. So a lot of this was actually developed initially at the Swiss Federal Institute of Technology. Now, when we returned to the United States, of course, NASA was interested in that, and they have supported a lot of this work. But almost all of the diagnostics applications actually came out of what was called the Defense Threat Reduction Agency, which was an organization which was worried about emerging biological threats and how you would detect them with rapid response. So since obviously there was COVID, which everybody knows about today, but there was also Zika and dengue, if you go back in time, even HIV. And so the reason why we're able to develop a COVID test in about 3 weeks was because we had all this DNA type chemistry developed that would allow us to do tests. Meanwhile, the poor Center for Disease Control, the CDC, was working with just 4-letter DNA. And I don't know if you remember February, March, and April of 2020, their tests were failing. And they were failing because of for lack of orthogonality, not to put too fine a point on it. So yeah, and then of course eventually the National Institutes of Health comes along, and it becomes sufficiently mainline that a lot of the technology that we now develop is going through the National Human Genome Research Institute, National Institutes of Allergies and Infectious Disease, and of course the National Institutes of General Medical Sciences. But then of course a lot of the other crazy stuff right now is being funded by the National Science Foundation. So they're doing the origins of life work here. So this is, I mean, for all of your listeners who hope to go into a career of science, right, the most important thing that we do here is try to persuade peers and various advisors to various institutions that that what we're doing is important. And so, for example, our life detection on Mars, the agnostic life finding, which has the fortunate initials ALF, as in that character from the 1990s television series. There's an Alpha Mars Society, alphamars.org, where you can go join, donate money, and actually get a part of the device development that we're going to try to persuade Elon Musk or the Blue Origins or somebody to put down on Mars to detect life on Mars.
Well, it seems like this technology is so valuable, especially when you're looking at space, right? I mean, radiation is something that is not only like cosmic radiation while traveling, but also cosmic radiation on the surface of Mars.
Yeah.
You know, I mean, if we're looking at colonizing other planets, being able to really understand, diagnose, and repair DNA seems like it's just paramount, because right now, if I remember correctly, like NASA is actually looking at, they're always looking at this, but they're looking at how they can minimize radiation exposure. They've looked at loading water around the space capsule to try and absorb some of that, and they're looking at alternative flight paths, and part of that is just to keep the radiation exposure below the recommended maximums, right? So there are lots and lots of things that go into this from just a cellular damage perspective, and what you're working with could help ameliorate or maybe even entirely correct that.
Yeah, I mean, I hate to say this to you, but your DNA is, you're not designed to do space travel. So, you know, if you go to Mars, well, there's a Robert Zubrin, who's head of the Mars Society, has written a book called Safe on Mars. Or I forget what the idea is. Yes, what you do is you have to worry about the proton storms that are coming out of the sun. So you put your groceries in a big donut, and when the proton storm is coming, you tip the donut towards the sun and let the groceries absorb the protons while you're sitting in the middle trying to protect yourself from these things. And as you mentioned, also, once you're on Mars, you don't have anywhere near the shielding that you have on Earth
Yeah.
by atmosphere. So you do have to dig and go down underground just to get yourself protected from the higher energy species. But absolutely, I mean, you are an organism, as you know, the Borg Queen said, you're an imperfect being designed by an imperfect being. So you have to worry about your fragile biology. In fact, this is one of the things that we really, I mean, what is intelligence? Well, you keep in mind that you are a generation 2 away from being able to understand your biology sufficient that you no longer have to suffer Darwinian evolution, which means having kids die to weed out by purifying selection the damaged DNA. So an intelligent life form is able to prevent that, right? Intelligent life form has enough command over their own biology that they don't have to kill kids in order to make up for the mistakes, to weed out the mistakes that are. Now we're close, right? But of course, once you're able to do that, you do have access to space travel as a biological life form. You really don't have access to much space travel without it.
Yeah, that's an excellent point. Well, and you'd mentioned the Borg, and right now there is this dichotomy in the sciences between the natural and the artificial, right? Between Watson and Crick's biology and Kurzweil and Moravec's artificial intelligence. Many futurists have asked whether machines will replace biology, but I'm wondering, does Hachimoji DNA tell us that past a certain point, this distinction may not matter?
Yeah. Well, I've always thought the Borg were a little stupid in not having figured out that honey attracts more flies than, a fly swatter. They really should not say resistance is futile at the outset. They should be a little bit more friendly, but that's the Borg. Yes. Keep in mind that you are, even in this conversation and everything you do, taking advantage of technology. without your suit and tie, you wouldn't be able to live on much of the planet. The sun would just burn the heck out of you. I mean, this is, and of course, going up to the northern latitudes, I mean, you were, as primates living at the Arctic Circle 45 million years ago in the Eocene, when it was 9 degrees warmer than it is today. So if you're worried about global warming, all you have to do is go back to the Eocene. It was much, much warmer, and you were living all over the place. And then, of course, the temperature of the Earth has gone down, 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 down to the ice ages. And of course, you basically left. You were forced out of everything except a very narrow tropical band, which includes Indonesia, where the orangutans are and stuff like that. But yeah, so you're now relying on technology that's transmitted from generation to generation by culture, not by DNA. You can argue that engineering is random Darwinian evolution, but really there is some rational way which we built a Boeing 747. We didn't do it by the random Darwinian process. And so you're actually taking much advantage of it. And I don't have a lot of fear of artificial intelligence. I don't see it as being anything more than supplementing what humans do and letting humans being able to survive. And I may be completely naive about this, but it seems to me that the dystopias that you see through so much of the current literature on the subject seem to me to be overblown. But there's no question that as a tool, Hachimoji DNA is an incredibly valuable tool that goes out from the macro, and even the flash drives are sort of macro, down to the molecular, and lets you do things that you could not otherwise do with molecular species that we know from natural biology, or even what we know from polymer science, fabrics, the other things.
Wonderful. Well, Stephen, again, it is truly a pleasure and an honor to have you with us today. Let me thank you so much for your time. And let me close by asking, what comes next for you, your research team, and for synthetic biology? Where should we be looking for this in the headlines next?
Well, I hate to make a lot of references to science fiction, but there's a book, a short story actually, by a guy named Theodore Sturgeon, who is of my vintage, called The Microcosmic God. I'm not sure if you've ever encountered this, but the idea is that we have a system that can evolve in the laboratory to do useful things. So one of the things that we have evolved in the laboratory is to create protein as analogs, antibody analogs actually, but these are made out of Hachimoji DNA that will bind to specific cancer cells and deliver drugs to specific cancer cells and to kill specific cancer cells specifically. And what's nice about this is it's very rapid turnaround. So we're looking at you as a potential patient. At some point, I hate to say this to you, but I'm hoping that it hasn't happened already, you will develop a tumor of some kind. Those tumors are personalized. Every tumor and every patient is in some sense different. The drugs, of course, are now generalized. They have, from everything from their development to regulatory approval, they have to be across a population. But we can conceive of following an autoimmune disease in you, a cancer in you in real time, to manage how it evolves as it is responding to therapy, treatment, and to keep ahead of it. And so as to personalize your therapy without anywhere near the great expense that some of these new some of these new cancer drugs are costing like $20,000 per dose. We can do it for $200 a dose with this evolvable system that we have under our control.
Amazing. Amazing. Let me thank you again so much for your time today, sir.
The pleasure is all mine.